Hello, everyone, and welcome to the second of our Fall Care Seminar Series speakers. My name is Laura Klepper. I'm the Associate Director for Education for the Center for Acoustics Research and Education. And today we are very excited to have with us Dr. Megan Ballard, who is a Senior Research Scientist from the Applied Research Laboratories at the University of Texas at Austin. Megan Ballard received her BS in Ocean Engineering from Florida Atlantic University in 2005 and a PhD in Acoustics from Pennsylvania State University in 2009. Ballard's research areas are related to underwater acoustic propagation modeling, geoacoustic inversion, and direct measurement of marine sediment properties. Ballard received the A.B. Wood Medal from the Institute of Acoustics in 2001, the R. Bruce Lindsay Award from the Acoustical Society of America in 2016, the Postdoctoral Special Research Award from the Office of Naval Research in 2011, and the National Defense Science and Engineering Graduate Fellowship Award in 2006. Ballard is a fellow of the Acoustical Society of America, the past chair of the Technical Committee on Underwater Acoustics of the Acoustical Society of America, and the editor for Proceedings of Meetings on Acoustics. So with that, we can all give a virtual warm welcome to Dr. Megan Ballard. Welcome, Megan.
Hi, and thank you very much for the introduction. So before I start my technical presentation, I thought I would just say a few words about myself, just to add to what Laura already gave in the introduction. So I am a senior research scientist at the Applied Research Laboratories, and I do a lot of experimental work, including at sea experiments. And the picture that I have on the slide right now is from our ARL Applied Research Lab homepage. And you can see I'm in the field gathering some pancake ice, and we had some acoustic probes that we were sampling the acoustic propagation through this like mushy ice layer. I usually spend, I would say, like 4 to 6 weeks at sea on a research vessel doing ocean acoustics research each year. And I do have a blog, which I can show you right here. And this is detailing some work we did in the Norse experiment, which was near Jan Mayen Island, north of Iceland. And it's called, you can see the webpage here, ARL in the Arctic.wordpress.com. Not all the experiments we've done are in the Arctic, but when we started this blog, that's what we were focused on. So if you want to see any more about my research, you can visit this blog. But the topic I'm going to talk about for you today is a 2-year record of acoustic remote sensing in Seagrass Meadow. And I want to acknowledge my co-authors, Kevin Lee, Andrew McNeese, Thomas Jerome, Robert Taylor, and Preston Wilson work with me at the Applied Research Laboratories. And then Ken Dunton and Kyle Capistrat-Fassa are at the Marine Science Institute, which is also part of the University of Texas at Austin. So to start off my talk, you may be wondering why we would be interested in studying seagrass ecosystems. And the reason for that is seagrass are very important. They provide a multitude of ecosystem services, and they also serve as important organic carbon stores. Our seagrass habitats are among the most threatened ecosystems on Earth. And for these reasons, long-term and continuous measurements of seagrass parameters are of primary importance for ecosystem health assessment and sustainable management. The next thing you're wondering is probably why we would use acoustic methods to assess and monitor seagrass. So the conventional measurement is using dissolved oxygen. It's made with a chemical sensor, but there's a problem because when the water becomes supersaturated with oxygen, free bubbles are formed on the seagrass leaves. And this is actually the typical condition in seagrass meadows. So as soon as the light levels are high, the free bubbles are formed, and then the dissolved oxygen measurements are underestimating the oxygen production. Some of you may know acoustic signals are very sensitive to the presence of bubbles, which cause dispersion, absorption, and scattering of sound. And so there's both the free bubbles that are floating in the water column when we have the supersaturated condition in the water, but then the seagrass leaves themselves also have gas channels within them, and these also cause the transmission loss. So I'm going to talk a little bit about previous work that's been done in seagrass ecosystems with acoustics, and then I'm going to move on and talk about an experiment we recently completed. So initial work in acoustic sensing and photosynthesis by seagrass was conducted by J.P. Hermand. He worked in the Mediterranean and he studied Posidonia oceana, which is a Mediterranean species of seagrass and has a very thick mat of interlaced roots and rhizomes that are entangled in the sediment. This is kind of a unique species of seagrass in the sediment root system can grow to be many meters thick. J.P. Hermand published several papers, but his main experiment result is shown in the box here, where he proved unequivocally that there are significant variations in the acoustic waveguide propagation characteristics, including the attenuation and time dispersion of the acoustic signals that were due to bubbles of photosynthetic oxygen production produced by the seagrass. Other work has been done in the Mediterranean species, both the Posidonia, which I previously described, and the Meadow species, which you can see in the picture in the middle of the slide here. These are a rhizome species that has these skinny leaves that come to the surface. So this work was done by Felsberto and his co-authors, and their main finding was that there's a significant production of oxygen bubbles in seagrass ecosystems that suggests net primary production is underestimated by methods that rely on the mass balance of dissolved O2 measurements alone. And then this work by Shang and his co-authors was conducted in Southeast Asia on the Dangsha Atoll. They looked at an intertidal species of seagrass called Seamedosa rotunda, and they found they could see diurnal variations of over 40 dB in their mid-frequency propagation. So I'm going to tell you about some of the work that my own research group has done. We used a very broad band acoustic excitation. It went from 1 to 100 kilohertz. And this wider band allowed us to identify the excess attenuation that occurred between 3.5 and 35 kilohertz when the bubble production was at a maximum. And so the figure that you see on the left is showing a comparison. If you look at the one labeled B, you can see the spectral levels between a bare patch, which was not vegetated by any seagrass, and then the seagrass at night, which is shown in the orange. And so you can see its received level is lower, and that's because of all the scattering from the seagrass leaves themselves. And then when you go in to midday, when bubble production is a maximum, and that's the gold line, and you can see there's some additional loss there, and that's from the bubbles produced by the photosynthetic oxygen production. This was our first work where we had a complete diurnal cycle of acoustic propagation in a seagrass meadow. And the way we did this is we took one of the small boats that you see in the photograph there, and we moored the boat in the seagrass meadow. It's only about 1 meter deep, and then we manned the boat for the entire 24 hours. And we borrowed a kayak from a neighbor, and we were able to go out and back to change personnel on the boat through the night. So this was continuing this type of propagation study, but now we have several diurnal cycles, and we compared measurements in the winter, and that's what you see on the left, to measurements in the summer, and that's what you see on the right. The top plot is a measure of solar irradiance, so that just tells us how much sunlight there is, and that's what's really driving the photosynthesis. We also measured wind speed, and then the acoustic measurements are shown on the bottom 3 plots. So the received energy level in the winter is what you see on the left here. And you can see it's a little bit lower when we have the storm come through. But then we have pretty high levels of received acoustic energy, regardless of what time of day it is. Although you do see some small dips here when we have more sunlight. The species of seagrass, when the winter comes, it is more dormant in the winter, and there's not as much biological activity. In September, here you can see the cleared diurnal sunlight cycles, and you can also see this clear decrease in the energy levels. And the 4 transducers were spaced between 1.5 and 12 meters away. In the spectrogram here too, you can see when the dissolved oxygen surpasses 100% saturation, then the free bubbles are formed and we get a decrease in transmission loss. And you can see that each day of the experiment. We did have some loss of data in the middle of the night here on September 19. We were using the computer to automatically record the data. The data is recorded every 10 minutes, but we got a Windows update in the middle of the night. Um, this was kind of a step forward from the single diurnal cycle because now we have almost a week of measurements. And the way we did this is we rented a house that was located on the bay. Then we cabled to the source and the receivers that were in the Seagrass meadow. And then we ran the cables back to the house that we rented. And so we're able to get these multiple diurnal cycles in that way. We still have all our recording equipment on shore for this experiment. This is just another analysis that we did with the seagrass measurements. In all this work, we're studying mostly Thalassia testudinum, although there are some other seagrasses that are sometimes interspaced within the seagrass. But it's a perennial seagrass and it grows from a long jointed rhizome, and the seagrass leaves are ribbon-like. In this work, we applied a Bayesian approach to estimate the posterior probability distributions for unknown model parameters. So we had our bubble size distribution that you see here, assuming Gaussian distributed bubbles. And then we really went after the void fraction of gas present in the seagrass meadow. We had a semi-bare patch. It was sparsely vegetated with seagrass. And for that case, we get a much lower void fraction than we do compared to when we made the measurements in the seagrass meadow. And you can see this clear separation between the two environments. And we repeated this experiment 5 times throughout the day in the seagrass meadow, and the void fraction remained high throughout the day. We also done a lot of laboratory measurements on seagrass leaves. This is one of the most recent laboratory experiments we completed, and we developed this new leaf model for the Thalassia testudinum that consists of a comparatively rigid epidermis, which is the outer shell of the seagrass leaf, and then on the center, this is the parenchyma. It's like a more mushy, compliant layer, and then these lacunae are the air channels that run the length of the seagrass leaves. We did acoustic resonator measurements and a combination of dynamic mechanical analysis along with finite element modeling to determine the acoustic parameters of the seagrass leaf. Okay, so I'm going to begin to talk about this main experiment that we just completed. In this work, we have an acoustic system that's deployed in a seagrass meadow. The water depth is about 1 meter deep. In this case, we have our electronics pressure housing located in the seagrass meadow, so everything's fully automated. We had an acoustic source and then 4 receivers between 1.5 and 12 meters away. We sent broadband chirps between 100 hertz and 100 kilohertz. The transmissions are every 10 minutes, every day and night for 2 whole years. We also collected ambient sound measurements from the first 30 seconds of each recording, and we also have environmental measurements at our experiment site, including water depth, temperature, and dissolved oxygen. And then we got supporting atmospheric measurements and solar irradiance. This is our experiment site. We're on the Texas coast near Port Aransas. This is a drone imagery. You can see these are 2 of my colleagues, Kevin Lee and Andrew McNeese, in the field, and our measurement area is outlined by these PVC poles here. We visit this area with small boats. We collect, this is someone here in the field collecting a core. We use this core to get an estimate of how much biomass is present in the seagrass. We made routine trips to Port Aransas to service our acoustic system every 2 to 3 months. This was a deployment between November and January, so kind of a lower productivity period, but still you can see we have a lot of growth on our pressure housings. And in fact, this salinity sensor here, we're never able to achieve good salinity measurements because the system is so biologically active, the sensor always became clogged. We were able to get good measurements of all the other environmental parameters we seek to get. When we service the system, we download all the data, we refresh all the batteries, and then we redeploy. We're typically able to do this within 24 to 48 hours, so we have a almost continuous measurement for the entire 2-year period. This shows all the data we collected. There's three different colored lines on this plot. The darkest line you can see here, we started the experiment in October of 2021. The middle green color is for 2022, and then the lightest green color is for 2023. And we made our final recovery of the equipment in the middle of November of 2023. As a whole, you can see we have seasonal patterns that are repeatable and then smaller diurnal fluctuations, and I'll talk about those for you in detail. The top plot here shows photosynthetically active radiation, which is known as PAR, and these are the wavelengths of sunlight that that plants use for photosynthesis. And you can see we have more intense sunlight as well as longer days in the warmer months. Water temperature, we have warmer waters between April and November. Wind speed measurements came from the nearby weather station at the airport about 3 miles away. We have calmer conditions between May and October. In the winter, we have more storms. We also got rainfall data from this weather station, and you can see often when we have a big rain event, there's also an increase in wind speed and then a drop in temperature. Water depth fluctuations are influenced by diurnal and seasonal variation in the tides, and then as well as big wind events, because we're in the shallow bay and the wind can actually push the water around significantly. These are the dissolved oxygen measurements. So generally you see levels building through the spring. They reach maximum daily values in the warmer months, and then they reduce to smaller fluctuations again in the fall. Now we're going on to the acoustic measurements. Here I'm showing you transmission loss. This was calculated from the pulse compressed signal on the hydrophone 6 meters away from the source. Actually, this is a hydrophone 1.5 meters from the source. You can clearly see the diurnal pattern of photosynthetic bubble production that causes increased transmission loss, with the highest levels occurring in the warmer months when the seagrass is most productive. You can also see a seasonal pattern in the nighttime hours that follows the life cycle of Thalassia testudinum, so that we have more transmission loss in the summer, as you can see highlighted by that orange line, and then less transmission loss in the nighttime hours in the winter when the seagrass are dormant. So our ambient sound levels here, I'm showing you the 25th ambient sound level, averaged over the 1 to 10 kilohertz band. And again, you can see the diurnal pattern in photosynthetic bubble production. This is associated with volumetric oscillations of the bubbles as they're released from the seagrass leaves. Can see it very clearly in the summer, but in the winter, it is often masked by wind events. And then we also collected above ground biomass, and this is reported as the dry weight of seagrass leaves, and this was collected using 15 centimeter cores. Just to show you the comparison between the kind of more dormant season in March, that you see that's what you see on the photograph on the left. So you can see there's smaller leaves. There's also some microalgae present in the photo there. And then if you look in September, here you can see the seagrass leaves are longer and the meadow is so dense such that you can't see the seafloor in between the leaves anymore. Okay, I showed you, this was the quick overview. Now I'm going to just show you a week-long period, and so we can get a closer look at the comparison between the different measurements. So it's the same data that I've shown on the previous slide, just looking at 1 week-long period from the month of May. And I've highlighted the times when we have sunlight and photosynthesis going on. So the maximum in PAR is well correlated with the maximum in dissolved oxygen, transmission loss, and ambient sound. That's what's highlighted by the orange bars. And you can also see every day we get an increase in transmission loss and in ambient sound at the time when the dissolved oxygen surpasses 100%. And that's what's shown by the black arrows. In 2023, there was a weather event, like a storm. It caused us not to have as much sunlight, so that you see that in the top panel. There was also some wind and rainfall overnight, and then the following day, the dissolved oxygen level does not surpass 100%, and then we do not see that increase in transmission loss or the increase in the ambient sound level. There you go, and that's highlighted by the black arrows. I also wanted to show you the effects of wind on ambient sound. We all know that wind is related to ambient sound. 2022, this weekend 2022 was slightly windier than the 2023 data, which are shown in the lighter green. And if you look at our ambient sound data, you can see the nighttime levels have higher ambient sound levels at night compared to the 2023 data, which are shown in the light green. Nevertheless, during the photosynthetic bubble production period, the dominant ambient sound is still the bubble production. Oh, of course, we know that rain can also affect ambient sound levels, and you can see there's some peaks in our ambient sound that is really well correlated with the rainfall measured by the weather station. So I showed you kind of all the data all at once, but it was averaged over frequency. So now we can look at the frequency dependence of TL. On each of these plots, the x-axis is time of day. It's UTC -5, which is local time with daylight savings. The x-axis is frequency, and this frequency goes from 1 kilohertz to 50 kilohertz. Although our chirp went up to 100 kilohertz, the transmission loss was so high such that we don't have good signal level above 50 kilohertz. You can see that there's an increase in transmission loss each day, even in our colder months. That's associated with the photosynthetic bubble production. And this occurs when the dissolved oxygen level, which is shown by the red line, this is the monthly averaged dissolved oxygen, when it goes above 100%. And you can see when we have the bigger excursions in dissolved oxygen, such as the month of July here, we have the most transmission loss. You can also look at the nighttime hours. Here you can see the frequency dependence of the transmission loss, and this is really attributed to the seagrass leaves. And there's more seagrass leaves in the summer months than there are in say, the winter months. So if you compare July and March. And yes, that's the seagrass leaf that causes the transmission loss. The transmission loss that we have through the seagrass leaves is consistent with having bubbles encapsulated. So it's at higher frequencies and lower levels compared to the free bubble loss. This is the monthly average ambient sound. So every day you can see we get a peak when the dissolved oxygen is above 100%, and it goes all the way from about 500 hertz to almost 20 kilohertz. You can see the sound peaks up, especially here in the later parts of the summer. One interesting point is the transmission loss here. If you look at the month of May, it really the ambient sound level really peaks at the beginning of the photosynthetic period, and then you can see these contours go down, have a negative slope here as we go into the afternoon. And I've overlaid the transmission loss data from the previous slide on this plot, and you can see that this decrease in the ambient sound level in the afternoon is associated with an increase in the transmission loss. So there's still as much bubble production going on in the late afternoon when we have this really high dissolved oxygen level, but the ambient sound level is lower because the transmission loss is higher. So our received level depends both on the sound generation and the propagation conditions. Finally, you can see this peak between 2 and 500 hertz, and it starts in April and persists through October, and it's kind of more in some months and less in others. And this is due to vocalizations from the spotted sea trout. This is kind of its mating call, and so it does this around sunset every day in the spring and late summer. So it's just an interesting feature we see in the ambient sound data. Okay, so we've seen kind of all the data band average at once. We've seen the data monthly average, so we could see its frequency dependence, but can we see its kind of everything all at once? And so for that, I've applied k-means clustering. So this sorts our acoustic data from an individual recording so it can be correlated and understood with sound generating processes. So k-means clustering, here I'm reading the gray box. We're trying to partition our in observations. So I do have a lot of observations because we record every 10 minutes for 2 years. And we sort them into K clusters, where K is a user-determined parameter. And I will sort both our transmission loss data and our ambient sound data both into 5 clusters each. So you can see from the kind of 2D illustration on the right here. First, you start with the data. You have these 4 points that are kind of randomly chosen, and then the algorithm iterates, and then at the end you can see there's 4 kind of clusters that make sense, where you see the data points are clustered and the dark gray dots are the data centroids or data prototypes, and they are representative of the data that are in that cluster. So I applied that to our acoustic data. The vector that we cluster over, the illustration here is just a 2D vector, but it's all the frequency bins of the spectra. So here I'll just go and show you the results. So on the left, we see the cluster centroids, and so they're normalized by removing the mean, but here you can see TL clusters 1 through 5, and then ambient sound clusters, AS, also 1 through 5. And then the normalizing factor is shown on the bottom left here. So we can see, for example, TL5 is the quietest cluster. Well, TL1 has more energy up here, and so it's kind of causing the most transmission loss. So the key thing here is to look at how they are as a function of time of year. So time of year is on the y-axis, and then time of day, which is local time, is shown on the x-axis here. Transmission loss is on the left, and ambient sound is on the right. Looking first at the transmission loss, I've overlaid the times of sunrise and sunset. And about 3 hours after sunrise each day, we start to see these light blue, dark blue, and green clusters. And these are the clusters associated with photosynthetic bubble production. Over in the second year, we again see these, but we perhaps see even more of clusters 2, 1, and then cluster 3 actually spreads to the overnight hours, and it's kind of straddling between probably 4 and 2, kind of spreading here. And then you can see we also have over the nighttime hours, so the times away from photosynthetic bubble production, we have TL5 and TL4. These are both kind of due to transmission loss from just the seagrass leaves themselves, where we have the cluster TL5 in the winter when the seagrass are dormant, and then we have more loss from cluster TL4 in the summer months. Well, we can do the same thing for the ambient sound data, so you can see the signal from photosynthetic bubble production in the clusters AS1 and AS2. Cluster AS3 is actually from those spotted sea trout vocalizations, and so that pulled out very strongly here. And then more windy conditions are shown in the darker yellow, and then the brighter yellow is calm conditions. So here I'm pointing with the red arrows. You can see just one example here where we would have expected possibly photosynthetic bubble production in the transmission loss and ambient sound, but we just don't see it. And the reason why is when we look at the PAR data, which is shown on the black, it's just a cloudy day. And we had one day where it was cloudy every morning, and there was also rainfall every morning. So we're able to separate these data out in that manner using this type of analysis. One more thing is that came out of this is this spotted sea trout vocalizations. There's about 40 to 50% more in 23 compared to 22. And in Texas in 2021, we had a major freeze event. You may have heard about it in the news because it was a week-long period where many people had very cold conditions and no power and no running water in their homes. On the coast, the water became so cold that almost all the fishes died. So they had a huge die-off of the fish population. And then it's been recovering. So we have more fishes in 22 and then even more fishes in 2023. And you can see that by the increased number of vocalizations. And you can also see some signal of more seagrass bubble production in the ambient sound measurements, where there's more light blue and dark blue in 23 compared to 22. And similarly, You can see as we move in the transmission loss data, there's more evidence of light and dark blue in 23 compared to 22. Okay, so now how can we use these data to understand more about the environment? So we've shown you now the acoustics are sensitive to bubble production and to biomass. And now the goal is like, how can we use these for ecosystem monitoring? So net ecosystem production in ecology, lymphology, and in oceanography is the difference between the gross primary production and the net ecosystem respiration. And so you can see that in this equation here. So what net ecosystem production describes the total carbon in ecosystem that is being stored, exported, or oxidized back into carbon dioxide gas. So the dissolved component of the gross primary production and respiration can be estimated from the rate of change of the DO measurements. And the goal of our current work is to estimate the non-dissolved contribution to gross primary production from the acoustic measurements. So we had this chamber that you see on the left here. This was deployed in the Seagrass meadow with this framed feet on the meadow floor. And then the funnel goes over the seagrass and it's out there for an entire day. And as the bubbles are being produced, they go into this collection chamber here. Then there's two pressure sensors. The trap pressure sensor is connected to the gas collection chamber, and then there's a reference sensor. And from the difference between these two, we can automatically monitor how much gas is being stored in the chamber. So the output of this is gas flux, and so that's a volumetric measurement per hour per area. We had two traps deployed next to each other, and they got very similar measurements that's shown by the dashed and dotted lines here. And then I have the mean of the two. And then the two colored lines, the orange and the greenish color here, are the ambient sound and TL measurements for the same day we had the traps out. So we use this data to create empirical regressions for gas flux between transmission loss and ambient sound, and you can see those regressions shown here below. So the traditional methods underestimate net ecosystem production until evolution is added. So we've done some preliminary calculations to show this. So the seagrass meadow is heterotrophic. That means it's releasing carbon to the atmosphere when we don't account for the evolution, the free bubbles that are produced by the seagrass. And then the seagrass meadow, when we do account for the bubbles, the seagrass meadow appears autotrophic. So that means that the flux here of oxygen is above zero. So this means we're sequestering carbon from the atmosphere. So we had a seemingly heterotrophic meadow with just dissolved oxygen, and then we account for the bubble production, and now we have an autotrophic meadow. So it does make a significant difference in the overall calculations. And we also can have some empirical regressions to account for above ground biomass, and that's what I'm showing on the figure here. This we use the nighttime TL, which I've traced out by this solid blue line right here. The total TL is shown by the light blue color in the background. We collected above ground biomass using cores like the one you see here. They're sieved in the field, and then the seagrass leaves are brought back to the lab where they're dried and then weighed and then normalized per unit area. We use the data on the graph here to make a regression for biomass, and that's shown by the equation below. The core data you can see has a lot of variability. The stars are individual cores that were measured on a particular day. And you can see for some cases, there's a wide variance between two cores collected in the same meadow on the same day, but widely different amounts of biomass present. So from this data, you can see there's more biomass in the summer than the winter. But when we look at the acoustic data, they trace out a more clear pattern where you can see in the winter it's dormant, and then about mid-April you can see there's a growth period that lasts about a month and a half. Then we kind of have maximum growth for about a month and a half, and then there's about a 4.5 month period where the seagrass are sort of decaying and dying, and we have a decrease in biomass. And then now we're in the second year and you can see the dormant period again. So the acoustic data provide a higher resolution with lower variance compared to the cores, which can be both temporally and spatially aliased because they're just point measurements. So this is some summary and some of our future work, and I do have some more slides about some of the ongoing work we're doing. So I just reported to you our multi-year record of sound propagation in a seagrass meadow. We investigated the frequency dependence and sensitivity to photosynthetic bubble production, as well as the seasonal changes in biomass. And the combined analysis of transmission loss and ambient sound are useful for interpreting the data. And you can recall I showed you, for example, that ambient sound plot where we saw the decrease in ambient sound in the late afternoon. That was caused by an increase in transmission loss. We have ongoing efforts to continue to analyze this data set, and we're doing some more modeling too. We're utilizing Bayesian inference techniques with a new effective medium model to estimate gas flux and biomass from our acoustic data. In future work, we'll investigate using passive acoustics for monitoring. So we're planning to deploy a network of acoustic nodes to investigate the spatial heterogeneity of the seagrass meadow and remotely sense CO2 uptake. This is a little bit about the Bayesian inference work that we're starting to do now. We take our seagrass environment and we're modeling it with just a 1D environment where we've got a water layover and then maybe some seagrass where there's perhaps a gradient in the density of the leaves as we get near the seabed, and then there's some sediment properties below that. We're using a well-known model for free bubbles in water developed by Commander and Prosperity in 1989, but for the seagrass leaves, which have their gas channels, the parenchyma, which is the soft tissue surrounding the gas channels, and then this outer shell, we have a new effective medium model for that. So that's described on this slide here. This is work done by Thomas Jerome, who's a postdoc in my group. He's developed 2 effective medium models and made some comparisons to data. One of them we call the micromechanical mass spring damper. And so the air channels are these gray blocks here, and then the squishy material, which represents the parenchyma, acts as some springy material separating the channels, and then this other one is just a cylindrical leaf with gas in the middle. And this may be more similar to a leaf like Syringodium, which we also have at some lesser extent in our Texas seagrass meadows. These are some low frequency resonator measurements that were used to create this effective leaf model, and we've made some comparisons to them here, and for this properties, they appear to fit the micromechanical model better. These parameters are determined according to the work we did in the lab to determine the properties of the seagrass leaf, but there is a loss factor that we're still pinning down. That's part of the model, but not something that we had previously measured in the laboratory. Yes. Ongoing work seeks to get more measurements over a brighter frequency band, and these measurements are being done in the lab using an artificial seagrass leaf. We can use the artificial seagrass leaves because we can isolate the substructure to compare them with the models. The natural seagrass leaves, the channels vary over the length of the leaf, and each leaf is different. So by using a 3D printer, we can construct leaves that have more uniform properties to make more straightforward comparisons with this new effective medium model. This 3D printer used this expanding thermoplastic polyurethane, TPU, and then this black outer layer is a thin rubber waterproofing layer. And that was applied after the 3D print. It's just like you buy it in a tube from Home Depot and it's applied to the leaves. So this 3D manufactured leaves were made by a different postdoc in my lab. His name is Colby Cushing. He has done both resonator measurements, and that's what I'm showing here. The seagrass leaves are these black bars that you see within this tube. There's a shaker source at the top, and then you get resonances in the tube. And from those resonances, you can back out the sound speed of the effective medium in the tube, which includes the water and the seagrass leaves. So here we're showing the resonator measurements comparing for first, the black is just water, the phase speed, and then the effective sound speed accounts for the waveguide parameters, which is the glass tube, which is not perfectly rigid because it has some elastic properties. So once that's all accounted for, you get the sound speed of the medium out. So for water, the sound speed ratio is 1, which is correct. For having 7 samples of seagrass leaves, you can see the effective sound speed is somewhat reduced. And then doubling the number of leaves to 14, you can see the sound speed ratio is reduced even more. The figure on the right here is the comparison to the 2 effective medium models here. You see we're getting better comparison to this MST effective medium model. High frequency measurements were also done through a phantom meadow of seagrass made up of different number of seagrass leaves. This source produces a plane wave, and then this hydrophone was moved through the meadow to scan these different ranges to look at propagation through the meadow. Different number of leaves were taken in and out, so you can see the measurements were made with 8 samples, 16 samples, and 24 samples. And in some cases, a row was removed, so you see different numbers of different peaks kind of as the hydrophone moves through a row of seagrass leaves. This is the when they're source only, measurements made as a function of range as this moves through. And then here you see when the seagrass leaves are there, and here we have 24 samples. This is a comparison of the 2 effective media models where the experiment is shown by the blue line here. And then here we're doing, and you can see the dash line from the micromechanical model provides a much better agreement with the data compared to the green line. And then looking at the furthest distance, creating the entire waveform, you can see there's very good agreement between the experiment and the micromechanical model, and then the cylindrical bubble simulation is shown by the green dash line, and again, it has too much loss. So really, the winner of this 2 models appears to be the micromechanical model, which is the one that had a model that was based more on our Thalassia seagrass leaf. Okay, so then now we've got the effective media model for the seagrass leaves and for the free bubbles, and now we're using that to do Bayesian inference to determine the seagrass leaves, how much biomass is there, and how much bubble production is going on. And then this is going to be compared with the bubble trap measurements and the biomass measurements, rather than using those direct measurements to create the empirical relationships. So they'll be used to check the Bayesian inference rather than to make the model. So here I'm comparing just, so we did have 4 receivers, as I said near the beginning of the talk. All the analysis that I showed at the beginning was from this closest receiver, which is 1.5 meters away from the source. And you can see the transmission loss increases as we move out further in range. The data here are for the receiver 6 meters away, and I'm showing data from January on the left, which is the winter when the seagrass are more dormant, and then data from August on the right. And then also comparing 2 different times of day, looking, these are times in UTC, but here you can see we've got more loss at 2100 because that's when we have more photosynthetic bubble production, and then comparing that to the middle of the night where we just have the seagrass leaves present. You can see there's more loss at night compared to during the day. And also if you compare these amplitudes, the relative amplitudes, we have much more energy coming in the winter than the summer. And in fact, if I plot these on the same scale, you can see very small amplitude coming in the summer. Looking at spectral levels here, you can see there's about 20 dB less energy coming in at night in the summer compared to the winter. And you can see we've got about an additional 40 dB during the photosynthetic bubble production period. Okay, so that's kind of our ongoing work, and we've also recently started a new project. This is sponsored by ARPA-E, which is the Advanced Research Projects Agency for Energy. They've spun off a new project called CCO2. And this was their press release, and ours was one of the 11 projects that was picked for this project. So here's our short blurb. We're developing an acoustic sensor network to quantify ecosystem activity and how effectively carbon is stored in shallow seagrass beds. Seagrass beds being an important sink for the coastal blue carbon cycle. Okay, so this is my last slide, and I just want to take a few minutes to thank the team that I worked on with this project. Ken Dutton is a professor of marine science at the Marine Science Institute in Port Aransas, which is part of UT Austin. And then Kyle Capistrant-Fossa is a graduate student in Ken's lab that worked on this project with us. Preston Wilson is a professor of mechanical engineering here at UT Austin and also has an appointment at the Applied Research Laboratories. Kevin Lee is a research scientist with me at the Applied Research Laboratories, and Andrew McNeese is our lead engineer in our group. And then Colby Cushing, Thomas Jerome, and Robert Taylor are three postdocs that worked on this project with us. So that takes me to the end of my presentation. And I'm happy to answer any questions we have.
Thank you so much, Megan. I think we'll all give you a virtual round of applause. You know, Megan, I've always loved how your work is just really pushing forward the role of acoustics in climate change research. I think it's been evident in everything that you've presented today. I love just the different approaches that you use. I think it's very creative on how you're incorporating acoustics and other ways to measure the environment. And I will also put in a plug that Megan is a guest editor for a Jazza special issue on, let me see if I can remember the title. I have it pulled up here. Climate Change, How the Sound of the Planet Reflects the Health of the Planet. So I have to put that plug in there for you, Megan. They're accepting manuscripts through the end of December. Is that right?
Yes, and I do really appreciate that plug. And I do want this special issue to be successful. We do need more papers. So if you're thinking about submitting and you have an idea, but you're worried about making that December deadline, please don't hesitate to reach out and contact me. There's always possibilities of extending deadlines and other things we can do. But this is climate change is real. It's happening to us. And as acousticians, we can really work together to help address this problem.
Agreed. And I think you're definitely helping to lead the way. So thank you, Megan. So at this point, I will open it up to questions. If anybody has a question, you can put a question in the chat, and I'm happy to read it out. If you would like to speak out loud, you can raise your hand, or if there's any questions in the room at UNH, please go ahead. It sounds like there might be a UNH question ready to roll.
This is Jen. I have a question. Well, I have a comment. First, hold on, you disappeared. Oh, there you are. Okay. First, Megan, I already turned in my article, so I'm 100% supportive of the special edition. And two, I might have missed it. I saw in your power spectra the frequencies that you were looking at for ambient sound ranged from 1 kilohertz to 100 kilohertz. When you had the graph a few slides before that, I think it was the orange line, and you had ambient sound on your axis. Was that over the same frequency band?
Yeah, we actually, our source did a kilohertz to 100 kilohertz, but in the end, we only processed up to 50 kilohertz for our transmission loss metrics because we didn't have good signal to noise ratio above 50 kilohertz.
Okay, so those ambient noise levels were from 1 to 50 kilohertz.
Okay, no, that was the transmission loss. And then the ambient sound, we actually didn't start this experiment thinking we were going to do an ambient sound study. But once we started looking at the data, we were like, oh, there is an ambient sound story here. So we had this system designed to really look at the TL. So it has like the source and the power amplifier in there. So there was some noise above 20 kilohertz. So we had some like tonal lines in the recordings at like 30 kilohertz, and it was not the quietest ambient sound system, but we saw a very clear ambient sound signal up to about 20 kilohertz. And so that's what we process for ambient sound in this paper. Since then, we've put out a number of sound traps, and we've started processing those up to higher frequencies. And so we can see some more higher frequency ambient sound data, but that is like still work in progress. We're not very far along in looking at those data yet, but we do have multiple years of high duty cycle single channel ambient sound recordings in the Seagrass Meadow.
That's awesome, and I hope that as you start looking at that ambient sound more, things like kurtosis or uniformity, I'm wondering if that's going to be even a better indicator for things that involve bubbles than just the amplitude.
Okay, yeah, that's a good suggestion.
All right, Andy, you have a question.
Hey, thanks for the presentation. It was super fascinating. I'm really interested in the technology and maybe like the logistics and the setup of this experiment, but I'm really curious with where the technology is at, and say it was 5 times cheaper, 5 times more expensive, or what would the experiment look like? Where do you want to take the research, and what's holding you back on the technical front?
So I did kind of talk a little bit about the stages we went in at the beginning of my talk. We're like, yeah, we were kayaking out to a small fishing boat at night, and then we cabled our system, and we, had all our stuff on to shore, and we could do multiple days. And then I kind of jumped up, and we had this whole 2-year thing where we had everything in pressure vessels. We worked with Jasco. You may be familiar with the Canadian company. They designed this, and you can actually now buy our system on their website. It's called the Ocean Echo. It has the signal generator, the amplifiers, and then all the recording electronics all in one pressure housing. And then the second pressure housing for us was just batteries. So I think the cost, we originally paid, we work with them, so we paid like the engineering cost to develop this. And I think it was like $90K, but then we bought the hydrophones and the source separately. The hydrophones are just HTI 90s, or no, they're HTIs. I don't remember. They're not 90s, they're higher frequency ones. And then the source is from Geospectrum. So we purchased a lot of this equipment, and but also work with the vendors to develop what we need. We do a lot of in-house building of electronics too, but for this experiment that I talk about here, we purchased a lot of this stuff.
Would you be looking to like make it better or expand spatially? Like what, I guess.
Okay, yeah. So the experiment that I mentioned at the end, the one that's we're being funded by ARPA-E to kind of do this kind of like spatial sampling, and it's really a carbon generation thing. For them, they really wanted it to be economically scalable, and it's based on the cost of carbon credits. So ARPA-E thinks we're moving to a carbon credit based economy. We're saying if you're like you're Google and you have a big data center, this also affects the environment quite a bit. So you need to use some of your money to purchase carbon credits to offset your carbon footprint. What is the cost of carbon credits going to be? Well, we can do this much more cheaply, we think, with passive acoustic sensors. So we're using Ocean Instruments sound traps. They make a 4-channel sound trap. And our plan is to do the passive acoustic sensing of the bubble production from the noise of the bubbles. And then we're also going to try to do a geoacoustic inference using ambient noise cross correlation. And that we're hoping we can determine the acoustic properties of the seabed. And then you may know Dr. Gabe Venegas, who's on the call with his hand up. He also worked in our group for some time before he went to UNH. And part of his work, he developed a regression between the compressional wave acoustic modulus and the amount of carbon in the seabed. And so we're hoping to leverage that in this future work to determine the amount of carbon stored in the seabed. So we have kind of two prongs with this, but the um, four channel sound trap is about $7,000. And then we also buy the hydrophones, but it's about an order of magnitude less expensive than the active system. And so not that we're done doing active acoustics, but in the next couple of years, we're going to focus on this passive acoustic experiment. The first phase of this project will use 6 nodes to look at area of the meadow. And then phase 2 uses 20 nodes. So we will have lots of data to process in the near future.
Super exciting. Thank you so much.
Gabe, would you like to ask your question?
Hi, Megan.
Hi, Gabe.
That was really interesting. I just saw that the paper that y'all just published came out. That was really exciting. And so I guess Ian, who's in the audience, I believe, and I and a master's student, we actually came up with, I think, the same idea at the same time, but we're deploying a measly 2-node system of sound traps in our own seagrass meadow up here, and we're trying to do sort of the 4-channel spatial passive acoustic monitoring as well as ambient sound correlation. And so where do you plan on doing this? Because we could maybe link up at some point, or if you don't have, I don't know if you have an eelgrass site picked up, but we have a lot of eelgrass up here.
Yeah, so we're partnering with Ken Dutton again for this next phase and going to go to our kind of Port Aransas spot because it's convenient. And in phase 1, we'll be collecting a lot of supporting data. We're going to do meter long cores again. All that stuff to get at like the kind of flux layer of carbon, and then once you get down below, you've got like the stored carbon that's really sequestered. So that's our spot. But for phase 2, we could potentially move somewhere else, and there's no intervention for a whole year in phase 2. So yeah, but we should definitely sync up and compare.
Speaking of syncing, the sound traps have quite a bit of drift in their clocks.
We've also looked at that. That's why we're doing the ambient sound cross correlation from the 4 channels, so they'll all be time synced, all in the same recorder.
I see. I
don't think you can do any kind of cross correlation with a single channel.
So we can talk offline about this, but we actually have a synchronization device between the sound traps. So we're going to be doing this sort of thing over 50 meters scale at this point.
Okay, yeah, let's talk more.
Yeah, all right, very good, thanks.
Well, Megan, I hear the bells chiming on campus.
Okay.
It means we're at the top of the hour. No, thank you so much for sharing your amazing science with us, and thank you for answering all of our questions. And I know Tony, did you have Tony's upset he didn't get a question. Sorry, Tony.
Well, if Megan has to go to class or teach or whatever, that's fine. But it's a word of caution from Megan and Gabe and whoever else is doing ambient noise measurements. Do you have time, Megan? Do you have a couple seconds? Yeah, so the mechanisms for ambient noise and for transmission loss, they overlap a little bit, but they're not exactly the same. So the ambient noise is when they release and they vibrate. Transmission loss, they don't have to release, and so you're seeing 2 different things.
Oh yeah,
so
for sure.
For sure, you know that. So the release is episodic. It depends on currents or when the bubbles get big enough, but it's not directly. It overlaps, but it's not the exact same thing as transmission loss. So I think you have the data, you have wind measurements, currents, tides. So I think you can tease apart that to some extent what that relationship is, but you're measuring 2 different things. But I think you can get at something else fundamental. If you're in shallow water and your meter of water in Port Aransas, those bubbles that release, they're big enough that they're going to make it to the surface. Everybody else stays on the leaves and redissolves. And so that's two different things. One stays and one leaves. And I think that's important. I'm not a biologist, ecologist, so I don't know why. But I think a little investment in those kind of two things about the difference between ambient noise and transmission loss might be worth pursuing, both for you and Kate.
Right, right. And that effective media model really just gets after the transmission loss portion. And the Bayesian inference that we're doing to start is all based on the transmission loss stuff. And then when we move into the passive acoustic monitoring with the cross correlation, that'll be different, a different approach.
Thank you for
Well, I'm
that.
done.
All right. Thank you again, Megan. This was wonderful. We really appreciate you sharing your time and your science with us. So thank you.
Yeah, thanks for having me.
All right. We will all say thank you and goodbye, everyone.